everybody, welcome back to the channel. Today we're going to be doing topic 6.3 of IB biology, which is called defense against infectious disease, but I like to just call it the immune system because essentially that is what it is. So with the immune system, there are three main types. There's the first line of defense, the second line of defense, and the third line of defense. That's how it's normally arranged. So starting off with the first line of defense, we call this actually the surface barriers. Surface barriers. And why do we call this the first line of defense? Because for any pathogen to engage with the second or third line of defense, it has to pass through the first line of defense. So for pathogens to enter the human body, it has to go through the skin. So that's one of the types of surfaces we have, skin. And what it does is actually protects the whole body in itself. And what you think is skin is not just on the outside of the body, but your body itself is like a donut, and the inside of your body, like your digestive tract, actually has mucous membranes, and this counts as an outer surface as well, right? So in terms of skin, we can think of it as like the super thick, hard stuff that's on the outside, and it also contains something called sebaceous glands, and that secretes something called sebum, right? Not sebum like the bodybuilder, but sebum, which contains chemicals and enzymes that kill microbes. Microbes are bad because they can enter the body and do all sorts of bad stuff. Um, they also have lactic acid and fatty acid. So acid types, lactic and fatty acid. So the pH is about 5.6 to 6.4. Oopsie. 5.6 to 6.4, which is in the acidic range, slightly acidic, and that prevents or reduces the microbial growth on the skin. Next, we also have mucous membranes. Mucous membranes. And they basically protect the internal structures. So like I said, the mucous membranes are on the inside and the cavities of the body. And what they do is they actually release fluids. So funny enough, actually, that's crazy. I just sneezed. I took a break for that. Sneezing is where those pathogens are actually released. So when you have substances, so your inside of your nose is also lined by mucosal membranes, and they have a layer of living cells which actually secrete a fluid, and this fluid actually washes away pathogens. So when you have snot, that's a lot of mucus being washed away. Um, also, this has acid, so mucous membranes also have a bit of acid that helps with removal of pathogens, along with your coughing and sneezing mechanisms. Next, we're going into blood clots. Blood clot sounds a lot like blood clot, which apparently isn't a word. If you guys have watched Weakest Link, leave a comment down below because that's what I'm referring to. Everybody's probably seen that. But clotting is something we call hemostasis, and it is where vessels, blood vessels, when there's like a lesion and there's bleeding. So let's say like you get a cut, right? And you're going to be bleeding. You don't want too much blood to be lost because blood loss is bad. And blood loss is bad, so we need to limit blood loss in a way, and we do that through clotting or hemostasis. And what we do is there's actually a process behind it that you'll learn more about. We have platelets, right? And what they do is they do something called primary hemostasis, and they just form a sticky plug and basically block that region of injury. So if you have, say, for example, a cut in your um, on your leg, it'll actually platelets will be able to put a sticky plug at the cut region so that blood won't ooze through. Next, we have fibrin, which forms a mesh trapping blood cells. So platelets are a sticky plug that prevents or closes that door that causes the blood to leave, whereas the fibrin is inside the house itself. So if we think of the body as a house and the blood as a person, the platelets would add a door, or if there are no doors, platelets would add a door. Okay, and fibrin, what it does is maybe puts in like barriers or furniture in the way of the person, not letting them in. If that's an analogy you want to think about. But there's something called a coagulation cascade. So what happens is when a blood vessel is damaged, so damaged blood vessel, it releases clotting factors, and clotting factors cause prothrombin to be converted to, wait, let me just put that there, prothrombin to thrombin, right? And thrombin causes conversion of to, fibrinogen to fibrin. And fibrin, again, is what forms a mesh. And the reason fibrin forms a mesh is because it's insoluble, whereas fibrinogen is soluble. Next, we have coronary thrombosis. It is when um, coronary, so to do with the heart blood vessels actually have blood clot formation, and the coronary arteries, especially because they have lots of pressure, those coronary arteries become blocked, and you have sort of build-up of these damaged areas or fatty deposits called atheromas, and these will then reduce the size of the blood vessel itself, right? So if you think of a blood vessel as being this tube, if you have like this build-up, right, due to clotting and other things, so not just clotting but also fatty deposits, then this area is reduced, right? Used to be like that. Used to be like that. And what do we know about arteries? High pressure. Because of high pressure, you can have rupturing. And what happens when you rupture a coronary artery? Well, coronary arteries provide the heart itself with oxygenated blood. Without that, the heart can't work, and without the heart, the entire body screws up. We'll learn more about that as we go into the high level topics, but for now, that's a baseline understanding of blood clots. Okay, moving on, we're going to the second line of defense, and this is called the innate immune system, which is non-specific. So innate, non-specific. Non-specific means all the pathogens are treated the same, and we have something called phagocytes. Phagocytes are cells which conduct phagocytosis, which is basically where blood cells, right, engulf a pathogen, right? They engulf a pathogen inside and break the pathogen down in something called a phagolysosome using enzymes called lysozyme. And that breakdown causes the pathogen to obviously go out. Some pathogen fragments are actually presented on the phagocyte itself. We call these pathogen fragments antigens, antibody generators. So antigens are presented on the surface of the pathogen itself. So like that. And these can be shown to antigen um, shown to T cells and other cells in the human body that will then create a specific immune response based on the antigens by creating antibodies. We'll go into that soon. Now, the third line of defense is the specific response, and it's unique to each pathogen. And we have something called lymphocytes. So there's B and T lymphocytes. And those lymphocytes are different from leukocytes, right? Because leukocytes are the whole class, right? These are leukocytes, and these are the lymphocytes. So they're a subcategory of leukocytes. And B lymphocytes are ones that actually produce antibodies, right? Antibodies, these are called plasma, right? And we also have memory B cells. And T cells, T lymphocytes, there are various classes. There's helper T cells, which activate B cells, and killer T cells, which kill off cells that are infected by a pathogen. So, for example, a human body cell becomes infected. Well, the human body cell won't be fighting for us anymore, as like for the human itself. It'll be on sides with the antigen, so it'll basically send signals for the killer T cells to actually um, kill the cell itself. So antigens um, have corresponding antibodies, and antigens are the surface parts of pathogens. And antibodies, what they can do, that antibodies can actually bind to these antigens, right? So if you imagine these are like the antigens on the surface of a pathogen, right? The antibodies, I'll do P for pathogen, okay? Antibodies can actually bind to these, and they can do several things. So there's stuff called opsonization. There's agglutination. So opsonization is when uh, actually that might not be relevant to this topic, but like agglutination, condensation, neutralization, those are some things we'll go into later in later topics. I was going to delve into like what antibodies does, but I don't think it's in my notes in terms of this topic. We'll go into it later though. If you do want to see a video on that, let me know in the comments. Next we're going into antibiotics. Antibiotics, as the name suggests, against, biotic means living things, and what they do is kill off mainly bacteria. And how do they do it? They target vital functions of these bacteria, such as the key enzymes in these bacteria, 7-S ribosomes, so enzymes, organelles that are only present in bacteria, so like 7-S ribosomes, like the cell walls, for example. So what it does is targets cell components that are not present in human cells, but pathogen cells, and it can kill those off. So for example, a bacteria, it'll kill off. There's two types, there's bactericidal and bacteriostatic. And bactericidal, cidal, it kills, and then bacteriostatic, think about static as pausing something, so it suppresses the ability of a bacteria to reproduce, or suppresses the bacteria's impact on the body, so it reduces the impact essentially. And viruses actually aren't alive because they don't contain the same things that are necessary for life to occur. Instead, they generally reproduce. People sometimes think of viruses as alive because they reproduce, move around, that stuff, but their reproduction generally happens from invading host material and stealing DNA, that kind of thing. So antibiotics can't actually hurt viruses, not so they can kill antibodies, but not viruses. And what do we need for viruses? We need antivirals, or the immune system itself, to form a way to tackle and tackle the virus themselves. And in terms of, let's go back into antibiotics now. There's antibiotics two types. There's narrow spectrum and broad spectrum. Narrow spectrum is specific, so it's like a sniper rifle, whereas a broad spectrum would be like a machine gun. It's just shooting everywhere. And you might think broad spectrum antibiotics are good because they can be used against a lot of bacteria. Why don't we just use them over and over again? Well, once you use antibiotics on a certain bacterial strain, sometimes just a few. So let's say these are all bacteria in a strain, right? And we're using an antibiotic, and it kills off this one, this one, this one, this one. But this blue one is living, right? Well, what happens in the next generation is, let me get rid of that. Happens in the next generation, the blue one's going to reproduce through binary fission, and when you have that, I had to sneeze again. When you have that proliferation of the resistant bacteria, so the new antibiotic that won't work anymore on those, so it won't work. It's not a possible option. So we call this antibiotic resistance. And when you gain resistance as a bacteria, it's really bad, which is why antibiotics are not being prescribed as much as they used to be. It's not good. And the type of antibiotic we know about is penicillin, and penicillin was discovered by Alexander Fleming, and it was when a petri dish had a penicillin like mold growing on it, and there was also like S. aureus on it, which is a bacterial infection, or bacteria. And when the penicillin was growing, um, the bacteria was suppressed, and because of that, he thought that okay, it's having an antibacterial impact, and he studied it but more. Turns out, yeah, it does. So penicillin and other antibiotics have become more frequently used in history. So you also need to know about some experiments that have occurred, and there's one about rats. I don't know, for the Florey chain experiment. So this is really simple to understand. So there were 8 rats, 4 of them, so they were all infected by rat. 8 rats all infected, 4 treated with antibiotic, 4 not, dead, survived. Bang, that's it. They also won a So Howard Florey, Ernst Chain, and Alexander Fleming all like won a joint a Nobel Prize. So that was also a cool thing I think you got to remember. Now we're going to go into HIV infection, and HIV stands for human immunodeficiency virus. And as the name suggests, it's virus and it leads to acquired immunodeficiency syndrome. And what does HIV do? It targets helper T cells, right? Helper T cells. What do we say helper T cells do? They activate B cells. And because helper T cells are damaged in this process, what happens is antibody counts are going to be lowered. Think about it. Lowering helper T cells means lower B cell activation, which means lower plasma B cells, which means lower antibodies. So what happens is you become more susceptible to other illnesses and diseases, leading to negative impacts on the body. The way HIV is transmitted: sexual contact, pregnancy, childbirth, breastfeeding, injections, blood transfusions, or other transplants of organs, and occupational exposure, which you can take notes on. Other than that, that's going to be it for this 6.3 video. I know it was a bit rushed again, like we're trying to do it in like 15 minutes, so that it's not too long for you guys to revise with. We didn't delve too deep into some topics because they're covered in HL. We'll go over that in the HL topics. Let me know what you guys want to see covered in the comments down below. Other than that, I'll see you guys in the next video. Peace.